Hello, namaste सबैजनालाई. So what we are gonna do today is we are gonna learn about cytokines. So we'll start with the definition. So what are cytokines? They are nothing but chemical substances that serve as messengers. Just like we message through our phones, they serve as messengers, mediating interaction and communication between various cells of immune system. So they are produced by broad range of cells, including immune cells like macrophages, beta lymphocytes, T lymphocytes, and mast cells, as well as endothelial cells, fibroblasts, and various stromal cells also. So a given cytokine may be produced by more than one type of cell. So for example, interleukin 1, there is not one specific cell that produces interleukin 1. So interleukin 1 can be produced by various cells. That's what this line tries to say. So to discuss about structure of cytokines, they are glycoproteins of molecular weight less than 30 kilodalton. That means they are very small, and their structure is alpha helix structure, and no beta plate structure is there in cytokines. So it belongs to one of four groups, that is hematopoietin family, interferon family, chemokine family, or tumor necrosis factor family. One particular cytokine may belong to one of these four groups. So what are the major classes of cytokines? The present nomenclature of cytokines includes all the compounds that were known earlier by various names, such as lymphokines, which are produced by lymphocytes, monokines, which are produced by monocytes and macrophages, interleukins, produced by WBCs, and they act on the same or different WBCs, and chemokines, which are involved in chemotaxis and other leukocyte behavior. So, properties of cytokines. Cytokines, they are comparable to growth factors and hormones in the fact that they act at very low concentration, that is in the amount of picomoles, and they act through specific receptors. So the common characters of growth factors, hormones, and cytokines becomes they act in very low concentration, that is concentration in terms of picomoles, and they act through specific receptors. While cytokines are different from growth factors and hormones in the fact that cytokines are inducible in contrast to growth factors, which are constitutive, meaning that under certain stimulus there will be higher production of cytokines, and without stimulus the production will be quite low. While in contrast, growth factors, they are constitutive, they are released in a certain concentration constitutively irrespective of instant stimulus. So hormones have mostly endocrine effect, while cytokines may be autocrine, paracrine, or endocrine. Now, unlike growth factors and hormones, which act mostly independently, cytokines can work together, and there are various interactions occurring between cytokines. The examples of interactions, the type of interactions being pleiotropy or redundancy effect. Pleiotropy refers to same cytokines having different actions on different target cells, whereas redundancy implies to different cytokines producing the same effect on the same target cell. So another property is synergy and antagonism. I mean, the type of interaction that occurs between cytokines is synergy and antagonism effect. So two cytokines may augment each other or help each other to increase the effect, that is synergy, or they may oppose each other in their actions, which is termed as antagonism or antagony. So another one is cascade effect. It refers to a series of effects mediated by different cytokines. One cytokine act on target cell to produce another cytokine, which in turn acts on another target cell, and so on and so on. So functions of some important cytokines. Though cytokines are secreted by a wide variety of cells, the major producers are helper T cells and macrophages. Cytokines produce a range of overlapping functions on the target cells or tissues, which can broadly be categorized into two groups. So, group 1 is promote development of cellular and humoral response of adaptive immunity. There is interferon gamma, cytokines such as interleukin 2, 4, 5. They promote the development of cellular and humoral response of adaptive immunity in our body. While another one is cytokines, they promote various responses of innate immunity. For example, induction of inflammatory response by interleukin 1, 8, tissue necrosis factor alpha, etc., and regulation of hematopoiesis by colony stimulating factors, interleukin 1, interleukin 3, interleukin 7, interleukin 9, 11, etc., antiviral activity by interferon alpha and beta, anti-tumor activity by tissue necrosis factor alpha and beta, pyrogenic activity. We all know tissue necrosis factor alpha, interleukin 1 and interleukin 6, they produce pyrogenic activity, meaning that they help in induction of fever. So, cytokines and diseases. So, pathogenesis of many diseases is characterized by increased expression of cytokines or their receptors. Common examples include the following: septic shock. So, septic shock due to gram-negative bacteria such as Escherichia coli, Neisseria meningitidis, etc., is mediated by endotoxins released by these bacteria. And these endotoxins, they stimulate macrophages to produce interleukin 1, tissue necrosis factor alpha, etc. Another one is toxic shock syndrome. It is caused by toxin released from Staphylococcus aureus, most commonly, which activates T cells non-specifically. That is, it is a superantigen. We will discuss what superantigens are in another video. For instance, superantigens are the one type of antigens that can activate T cells non-specifically, leading to massive cytokine release, and that in turn activates macrophages to release large quantities of interleukin 1 and tissue necrosis factor alpha. So another one is cancers. In several malignancies have been associated with increased level of interleukin 6. Example, cervical cancer, bladder cancer, etc. And Chagas disease, which is caused by a parasite named Trypanosoma cruzi, is shown to inhibit interleukin 2 alpha receptor, thus blocking the interleukin 2 action, which leads to inhibition of helper T1 activity and thus immunosuppression. Now, another very important term is cytokine storm. It is a condition where the cytokines are produced in excess, leading to hypercytokinemia, which can cause significant damage to the body tissues and organs. Normally, the production of cytokines is kept in check by body. However, in some instances, the reaction becomes uncontrolled and too many immune cells are activated in a single place. This precise reason for this is not entirely understood, but may occur in a number of infectious and non-infectious diseases, including graft versus host disease, acute respiratory distress syndrome, sepsis, Ebola, avian influenza, smallpox, systemic inflammatory response, COVID-19, etc. So, what are the uses of cytokines? Are there any uses though? So, interferon gamma, it has been used in the treatment of hepatitis B, hepatitis C, hairy cell leukemia, multiple myeloma, and chronic myeloid leukemia. Interferon B, it has been used in the treatment of multiple sclerosis. Similarly, interferon gamma in the treatment of chronic granulomatous diseases. And cytokine toxin conjugate, it has been used for target cell destruction. So cytokine, what it does is it helps in binding to the target cell and the toxin destroys the target cell. So this is the end of the slide, and thank you so much for bearing with me and learning about these beautiful mysterious chemical compounds called cytokines. Thank you. We'll see you in the next video.